welcome everyone to this edition of the Cameroon Town Hall for Healthcare Professionals. Today is going to be a very interesting session. We're having one of our regulars, Dr. Kenji Obadia. He's going to be our guest speaker today. We still have 10 people who have joined. I will give some few minutes for others to join. In the meantime, we can just, I think, say hello to some of our fervent participants of the town hall. I can see Franklin Maloba joined already, Tanya Bless, Simon Joya. And of course, today we're going to have Dr. Matt Sandrine Polo, who is going to co-moderate the session with me. So she's going to co-moderate in French. Hello, Matt. Welcome.
Hello, Elvis. Welcome to all. Merci d'être là. Aujourd'hui, j'ai le plaisir de joindre la session et de modérer en français. Et j'ai aussi le plaisir d'assister à la suite de la conversation qu'on a commencée la semaine dernière sur la médecine de précision. Donc, j'ai hâte d'entendre Dr. Kenji. Merci.
Thank you very much. And it's really going to be interesting to have you co-moderate with me. And of course, we have our chair, Dr. Muno. Hello, Muno. How was your week?
Oh, my week was great. And thank you, Elvis. And I just want to also thank Kenji for being available in order for us to continue the discussion on precision medicine and companion diagnostics. So I think this is a field where the science is going towards, and evolution in medicine and also in the field of biomedical science will make a great difference. And I hope during giving these discussions we're having, we can, in West Africa, I don't love to put Africa in the same basket. In West Africa, in Central Africa, in Cameroon, we can catch up and hopefully in the next few years be at the same level as the US. That's my hope always.
Really interesting. And companion diagnostics, certainly a very important part of medicine as we continue to advance in medicine and trying to be as precise as possible in the therapy that patients receive. And of course, we would not have had an expert, another expert to take us through this topic other than Kenji, who is a trusted authority in the space of diagnostics. So Dr. Kenji, thank you very much for accepting to present this session. We have some few people I would like to say hello to. I see Clarence. Hello, Dr. Clarence.
Hello.
Good to have you. How is it in Germany today?
Everything is fine, thanks. Everything's fine.
Wonderful. It's always good to have you. We have Dr. Ekiti Martin. Hello, Dr. Ekiti.
Hi, everyone. I am looking very much forward. This is one of the only sessions that I have no idea, reading the topic, what is going to be about. So I'm really looking forward to learning a lot from this.
Ekiti Martin, you are not alone. When they brought up the topic in our forum, moderators forum, I asked that companion diagnostic is whose business? Is it Kenji's new business he has incorporated and has not told me? And then Mono said, no, it's not a business. It's a form of diagnosis. I had to go start reading about what companion diagnostic is all about. So you are not alone. So it's quite interesting. Of course, we have Franklin Maloba. Hello, Franklin. Welcome.
Hi, Senior Darcy. Yeah, good morning. And it's a pleasure to be in the town hall once more. And it's a pleasure to learn about companion diagnostic as somebody for the medical laboratory fee. Thank you. I'm more to that from an expert, you know.
It's always good to have you on the session. Thank you so much.
Thank you. Thank you too. And thank the town hall for doing this.
Yeah. And of course, we have another person with a lab background, Dr. Forty Luis. Dr. Forty Luis, it's good to see you. How is it in Canada today?
No, it's great. Great. I'm so pleased to be part of this presentation today. I'm looking forward to what was like a dream back home when we used to discuss on this topic, especially with Kenji. I think you understand what I mean. So it's like a reality come true. So
wow, that's
yeah, looking forward to the presentation.
Great. Ladies and gentlemen, we are now 18 of us on the session. If you are joining for the first time, this is the Cameroon Town Hall for Healthcare Professionals. We meet every 2 weeks to discuss on health topics that impact the lives of our brothers and sisters back home, and we share knowledge, both best practices from different parts of the country. We are now in about 36 countries. There are people who have participated in this session, and I think it's a really knowledge-sharing platform. Everybody in the town hall is equal. We all try to be respectful of each other each time we discuss, and especially during the question and answer session. So we always start with the presenter. He would have 1 hour to do his presentation, and then we go into the question and answer session, where you feel free to ask any question that you might have noted down during his presentation. I'll hand over to Dr. Sandrine to introduce the session again once more in French.
Thank you, Alice. Bonjour tout le monde. Alors, aujourd'hui, nous avons un sujet très important sur le diagnostic avec de précision. On va demander à tout le monde de réserver leurs questions pour la fin et l'inviter aujourd'hui à 1 heure de présentation. Bonne réunion.
Merci beaucoup, Sandrine. And now I have the pleasure to ask our chair, Dr. Bono, to a brief presentation of the speaker. I know most of you know him, but just for those who might be joining for the first time.
Well, Elvis, and I just want to thank Dr. Kenji and for accepting our short notice for us to continue this discussion on companion diagnostic and precision medicine or personalized medicine. There is no one best to talk about this other than Kenji. I can introduce Kenji for a whole week. And what we need to know is that Kenji is very well qualified. He's right now the manager of infectious disease and exotic testing at Standford Healthcare System. And he has a ton of experience. He's an expert, a consultant with WHO, and has driven healthcare in Cameroon and has done a lot of research right from the days back in the days in Cameroon, and has been involved in shaping the infectious disease platforms in Cameroon, research on different organisms such as malaria. He's an expert in what happens in Cameroon. He understands the healthcare system of Cameroon. It may be better than most of us here and Stanford, and has been there for more than 4 years and has driven the lab to the best of technology you can imagine. So, Dr. Kenji, we are very pleased to have you here in the town hall. You're a regular, and you've been involved since the beginning and the conception of the town hall, one of the founding fathers of this town hall that we're doing today. So thank you for once more accepting our invitation. So I can go on and on and on, but at least let's keep it this short, brief. I know you don't want us to get to the point of introducing you all day. So Elvis, over to you.
Thank you very much, Dr. Mono. Of course, Dr. Kenji is a trusted authority when it comes to this field, so we would have the opportunity to listen to him. So over to you, Dr. Kenji.
Well, thank you very much. Thank you, Elvis. Please, can you hear me okay? I'm in a hotel and the internet has been a little shaky, but can you hear me okay? All right, so Elvis, please, at any time, if the internet kind of breaks, just shoot me a message on WhatsApp. Then I can try to switch the connections. So thanks, everybody. It's once more a very great pleasure for me to be here to talk to you today about companion diagnostics. So today we'll explore companion diagnostics, which is a component in the evolution of personalized medicine. As we know, personalized medicine, it really aims to tailor treatment to the individual characteristics of each patient, whether through their genetic profile, it could be through biomarkers or other key indicators. And we'll talk of companion diagnostics, right? Look at a companion, right? So it's actually tools, right, that makes this precision medicine possible. So they allow us, to identify which patient are likely to benefit from a specific therapy. And, I'm going to talk more about it. And also it enables us to predict adverse reactions and help clinicians, to choose the most effective and safest doses for each individual. So in this presentation, we will take a look at how companion diagnostics, are driving advancement in healthcare, transforming, what we know as traditional medical practice and making treatment more targeted, effective, and safer for patients. Before I dive any further, I just want to say whatever I'm saying here is actually my own opinion, and it doesn't represent the stand of any organization that I consult for or that I work for. And I'm here as Kenji Obadia. I don't represent any of the organizations that I work for in this presentation. So moving to the next slide. I really want to start with this quote from President Obama. So in this quote, President Obama captures the essence of personalized medicine and its potential to actually revolutionize healthcare. So if you look at the quote, he actually begins by acknowledging that doctors have always understood that every patient is unique, and they have strived to tailor, treatment to individual needs. An important historical example that he uses, which I was, really taken in awe of the example that he uses, is blood transfusion, right? For blood transfusion, you realize that you need to match the blood type, right, to the particular patient that you want to give it to. So there was a breakthrough in medicine. Then President Obama, he then asks a very provocative question, that what if matching a cancer treatment to our genetic code was as easy and standard as matching blood types? So this vision actually captures the heart of personalized medicine, which uses our genetic information to, to tailor cancer treatment and other therapies to the specific biology. And finally, he makes the vision even more accessible by asking, what if finding the right dose of medicine was as simple as taking our temperature, right? And this really emphasizes the goal of making personalized medicine routine, simple, and widely available in healthcare. So President Obama's quote actually encapsulates the promise of personalized medicine, making healthcare more precise, effective, and individualized for each patient. So I'm going to start by talking about personalized medicine in brief, then I'm going to then narrow it down to companion diagnostics. So to understand personalized medicine, let us take a look at how two major institutions define it. The first is the President's Council of Advisors on Science and Technology. They define personalized medicine as the tailoring of medical treatment to specific characteristics of each patient. Importantly, this definition clarifies that personalized medicine doesn't mean, right? It doesn't mean that you are creating unique drugs or devices for each individual. So instead, personalized medicine involves, classifying patients into subpopulations, and these subpopulations might be more susceptible to specific diseases or more responsive to particular treatment. So in contrast, the National Cancer Institution expands on this by emphasizing, the role of genetics and other factors. So the National Cancer Institution defines personalized medicine as a form of medicine that uses information about a person's gene. It could be information about a person's protein and the environment to prevent, diagnose, and treat diseases. So in essence, both definitions, they kind of highlight that personalized medicine is about tailoring care to an individual's unique biology and environment, right? Whereas it is more accurate diagnosis and targeted treatment or even preventive measures as well. So let's look at this graphic. I really like this graphic because it kind of shows us, the before and the after. So this graph illustrates the fundamental difference between the traditional one-dose fit-all approach and the more tailored personalized medicine approach, which is guided by genomics and pharmacogenomics. So on the left hand here, we see that the traditional method where every individual, regardless of their genetic difference, receive the same dose of drug, right, as you can see here. So in this case, it is 100 milligram. So this one-size-fit-all approach assumes that all patients will metabolize and respond to the drug in the same way. However, this often results in variability of outcome, right? Some patients may experience adverse effects, while others may not receive enough of the therapeutic benefit. But let's take a look at the right? And you'll see several subgroups here to my right. So on the right side, you see a more personalized medicine approach wherein treatment is adjusted based on the patient's genetic makeup and a specific ability, right, to be able to metabolize the drug. So you see here, patients are grouped into 4 categories, right, onto the right. The first one you have is ultra-rapid metabolites. So these are individuals that metabolize the drug very quickly, so they might require a higher dose, right? For example, here, this individual is getting 500 milligrams to be able to achieve that therapeutic effect. Then you have the extensive metabolizers. So this group, they metabolize drug at a normal rate. So they stick with the standard 100 milligram dose. Then you have the intermediate metabolizer. So these individuals, they process the drugs more slowly. So they require a lower dose, perhaps somewhere around 50 milligrams to avoid toxicity. Then you have the poor metabolizer. So these are individuals that metabolize the drug very slowly, and they may need even a lower dose, such as maybe 10, micrograms to avoid adverse effects. So by leveraging genomic data, we can move beyond this one-size-fits-all approach model and ensure that each patient receives the right dose based on their genetic profile. And this approach not only optimizes the efficiency, but it also minimizes the risks of side effects, leading to better clinical outcomes. So these are some associated definitions that we are going to look throughout the presentation. So let's just cover 2 of them, or actually 3 of them. First will be genomics. So for genomics, it just refers to the study of the entire genetic makeup of an individual. It kind of helps us understand how genes influence health, development, and disease. And another key term that I want us to look at is what is referred to as pharmacogenomics. Pharmacogenomics, it looks at how our genetic makeup impacts our response to medication. So by analyzing we'll talk of pharmacogenomics. So it's actually a field of science. It combines some pharmacology and genomics to study how an individual's genetic makeup will affect their response to drug. And the last thing I want us to look at in terms of definition before we proceed is single nucleotide polymorphism, because you can't really talk about personalized medicine without talking about single nucleotide polymorphisms. So these are small genetic variants, and they are single nucleotide variants. So each SNV reflects a difference in a single nucleotide letter. For example, if you look at these 3 individuals, you see that the difference between these 3 people is just, single nucleotides here. The rest of the genetic makeup is the same. In as much as it's just a single nucleotide difference, the difference between this SNPs expression may actually modify a drug's therapeutic response or adverse effects. So that's why the genetic makeup is very important to take a look at. So personalized medicine works through several key mechanisms. It starts first with genomics and genetic testing, where DNA sequences actually helps identify genetic mutations. Then pharmacogenomics helps predict how a patient will respond to certain drugs, based on their genetic profile. While you now move to the next biomarker identification, is going to allow for a more precise therapies by targeting specific disease markers. Then we have we also have fields like epigenomics, which study gene expression and changes, and transcriptomics, which focus on the RNA part. You also have proteomics that is also involved, and metabolomics as well, which further expands, our understanding by analyzing proteins and metabolites. Then you also have the microbiome as well, analysis, which gives an insight as to how our gut bacteria influence our health and treatment. And beyond that, you have immunogenomics, which is crucial for immunotherapy. And again, in this era, you also have artificial intelligence, which helps to analyze the vast amount of data, to personalize the treatment. And liquid biopsy also allows for real-time cancer monitoring. Then companion diagnosis ensure that patients actually receive the most effective personalized therapy. So these are some of the benefits of personalized medicine. It brings numerous advantages. First, by tailoring treatment to each patient's genetic profile, we see enhanced drug efficacy and improved safety. Patients receive medications that are more likely to work for them while minimizing the risks of adverse effects. Secondly, it leads to improved diagnostic accuracy, helping doctors actually classify diseases more precisely and catch them earlier. So this targeted approach actually contributes to cost effectiveness, as it reduces the need for what we usually call try and error prescribing. So, and most patients too is going to lead to more favorable clinical outcomes. Of course, it still has its own limitations. Especially in the US here, reimbursement is still an issue, when it comes to it. There's still some ethical issues too regarding genetic testing and data sharing, and just integration and a host of other things that can. So now let's dive into companion diagnostic. So when you talk of companion diagnostic, what is it about? So it can either be an in vitro diagnostic tool. So this would be like the laboratory tests that we typically run in the lab, which could be, using next generation sequencing. It could still be an imaging device. And what it does is that it provides critical information that is required for the safe and effective use of a corresponding therapeutic treatment. Now, so companion diagnostic is not in isolation, right? So you have a test that goes alongside a particular medication. So for those that really wanted to know a little bit more why it is called companion diagnosis. So something has to accompany it, right? And companion diagnostics play an essential role in personalized medicine, as I highlighted before, by identifying responders. So these are patients that are likely to benefit from specific therapy. And it can also help predict risk. So these are patients that are at high risk of adverse reactions to a particular drug. And it can also help optimize dosing for patients. And I'm going to talk more about that as we proceed. So the Food and Drug Administration has cleared and approved several companion diagnostic devices. So this is the link to a site where, if you want to know which particular, cancer type. So you start with the gene, you look at the alteration, the cancer type, then the drug, then which companion diagnostic goes with that drug, right? So this is a very good tool for medical doctors. For example, if you want to prescribe, maybe a particular drug, it could be olaparib, you could check which companion diagnostic can go for that one. You see that Foundation one has a companion diagnostic test using next generation sequencing, and the specimen type is fresh frozen plasma that you can use in that case. So I'm just going to take one of them just to talk about it here. And the one I'm going to pick up is the one that is developed by Roche. So it's called COBA's EGFR mutation test. Now they are in version 2. So there are a few things that I would like to highlight about this test. So when you go to that site and you click on the test, you may be able to get some of this information. So the purpose of this particular companion diagnostic test is to detect about 42 mutations in the EGFR gene, and it targets several exons. There are, I think, 4 different exons. And these mutations, they are associated with either sensitivity or resistant to EGFR targeted therapy in non-small cell carcinomas. So that is why it is called a companion diagnosis, because you're going to do it and you're going to say if that patient is sensitive or resistant to those particular targeted therapies. And the sample types for this particular test, you can use fresh frozen plasma. FFPE, not fresh frozen plasma, it's tissue. Then you can use plasma as well. So it actually makes it versatile for when you're talking about liquid biopsy. And what's the relevance, right? What is the relevance of this test? Is going to help guide treatment decision, especially for tyrosine kinase inhibitors, and also monitor if that patient has acquired resistance, right? Such as the T790M mutation. And the turnaround time is pretty good. So within 4 to 8 hours, you are able to get your results. And again, this is through PCR, right? This is not sequencing. So you are able to run this test and you can actually see if a patient is able to respond to a particular treatment or not. Now, I've kind of created this table. It's a little busy, so I kind of summarize what I would say the most popular laboratory techniques that I use to develop companion diagnosis, right? And I'm just going to rush through them. Then at the tail end, I'm just going to focus on one, which is next generation sequencing with a case study. So the first technique that I want to talk about is PCR. Of course, due to COVID, PCR became extremely very, very popular. And the reason why I'm bringing these techniques is that we have people here that are laboratory scientists that will use these in the lab. We have doctors that will want to request a particular test for an individual. So it's good to just have an idea, which test does what. Again, not every test is created the same. So PCR, what it does is that it amplifies a specific DNA sequence. So for PCR, you need to already know what you are looking for, right? You can't just go blindly for PCR, right? And that's one of the limitations of PCR, because you cannot detect what you don't know. So you have a specific gene sequence, right, that is going to detect a specific mutation. And one of the advantages is that it's a very familiar workflow. It is highly sensitive and it is highly specific. Then you have another alteration of PCR, which is called droplet digital PCR. This one has, a higher sensitivity, especially if you are looking at very low frequency variants. So these are variants that are not highly expressed. That is where you want to use digital droplet PCR. Then we have FISH, which is known as fluorescent in situ hybridization. So here you kind of use fluorescent probes to be able to detect specific DNA and RNA sequences in a tissue. And it's really best when you want to look at rearrangements of genes. And for example, if you are looking at the HER2 gene or the LK gene as well. The disadvantage with FISH is that it is labor, intensive. It requires specialized equipment and all the like. They also have immunohistochemistry. It is a very simple and widely used technique, but the problem with immunohistochemistry is that the interpretation can be semi-quantitative, and at times it could be subjective. And of course, next generation sequencing, which is the elephant in the house, right? It is high throughput sequencing. And the advantage with next generation sequencing is that you can detect what you don't even know, right? So it is high throughput. You can, detect multiple mutations, right? One of the tests that I'm going to talk about, you are able to detect like 550 different genes, right, with next generation sequencing. But you cannot do that with PCR, right? PCR can only do so much in terms of multiplexing. And you also have another technique, which is called microarray, right, which you can use for gene profiling. So between all these techniques, the ones that are so popular that I've seen being used is PCR and next generation sequencing, which I just wanted to hone on both a little bit. So as I said previously, PCR is a familiar workflow. Access to equipment is available, especially after COVID, right? Most of the countries have these PCR equipments that are sitting there. Then next generation sequencing, as I said, it's you can it has a very high discovery power, right? So you are able to look at, a lot of genes, higher mutation resolutions that you cannot get if you are using a PCR. And again, it is a massive parallel, and you can detect gene expression changes down to about 10%. But they all have their own challenges as well, starting with next generation sequencing. As you say, you use it to probe several genes. So if you are just looking at 1 or 2 genes, you don't really need next generation sequencing that much, right? It's not cost effective. Let me not say you don't need it. It's not cost effective. You are just looking at a few genes, right? That you could still use PCR for those. So that's one of the drawbacks for next generation sequencing. Again, cost too, is a big challenge when it comes to next generation sequencing. And I already gave some limitations for PCR, right? The fact that you can only do so much multiplexing. So I know the previous town halls, I know Dr. Fuma talked a lot about next generation sequencing. Other people talked about it. I'm just going to hone down on next generation sequencing just a little bit. Then I'm now going to give one case study on how we have used next generation sequencing for companion diagnostics and how it is helping patients. So when you talk of genomic sequencing, what do you actually mean, right? So it is a process where you want to determine the nature, and not only the nature, but also the order of the 4 nucleic acid bases. And when we talk of the 4 nucleic acid bases, we are referring to the A, T, G, and C. So during sequencing, what happens is that you have all this information, right? It is converted into data, then it is then analyzed, right? Then you have, all the information there after analysis. So sequencing has actually evolved, right? Now we are referring to them as generations, right? When sequences started, we had what was called Sanger sequencing. I mean, people still use it, even at Stanford, we still use Sanger sequencing, but it's kind of sunsetting. Then we move further to what is called second generation high throughput sequencing. So this is where Illumina will come in and all the like. Then we now have the long generation, right, where Oxford Nanopore comes in, and this one they mostly do, long reads and a host of other things. So these are the generations, and these are how things have evolved over time. Just to put it in perspective, next generation sequencing evolved in about 2006, and you can imagine that to sequence the first human genome took about 13 years, right? And an estimated cost for the first initial draft was about $300 million to sequence the first human genome. Whereas now we can sequence a genome in a day for less than $1,000. So things have really changed, and sequencing has become, more and more available and affordable. And these are the different next generation sequencing technologies that are available out there. You have sequences by synthesis by Illumina. I won't go so much into all those details. So when you talk of next generation sequencing, what do we actually do in the laboratory when we talk of sequencing, right? So this is just a summary slide. So what happens is that we get the samples in the laboratory, we do some pre-processing, right? It could be extracting the DNA and all the like. We move to what is called library preparation in the laboratory, where you want to add some adapters. Then we now move to sequence that sample. Now, when you are done sequencing, you can imagine a lot of data is being generated after sequencing. So you need to have all the bioinformatics pipeline to be able to take that data, do all the data QC, but most importantly, making sense out of that data, right? You need to be able to interpret the information from that data as well. So in a nutshell, this is some kind of an overview workflow of next generation sequencing. These are some platforms that are available out there in terms, I mean, if you are lab or if you are considering implementing next generation sequencing for companion diagnosis, Illumina is like the giant in the industry. I worked for Illumina for 4 years. I'm not in any way endorsing any of the products, but I mean, it's just left for you to see which one works best for you. So Illumina offers a plethora of different types of sequencing, depending on the scale of what you want to do, and I've summarized them there. Then again, using Illumina, it has its own advantages and disadvantages. You can take this slide and see some of those advantages and disadvantages if you want to use Illumina. But I wanted to talk a little bit about Oxford Nanopore, because I think this is really changing the game in terms of sequencing, right? So before, we're used to all these giant, sequencers, but now Oxford Nanopore has been able to produce a sequencer that is just like an iPad. And they even have one that is just like a mobile phone. So you can actually deploy it to the field, right? And actually do your sequencing in the village. They are just using, this Oxford Nanopore technology. So I'm just highlighting these, for those that are interested in implementing things like this, that these things are available out there. And again, ONT has its own advantages and limitations as well. All right, now to the core of my presentation, I just want to take one case study where you can actually see how the lab or how companion diagnosis fits into the realms of personalized medicine. And I'm going to talk about TrueSight Oncology. TrueSight Oncology is developed by Illumina. I had the opportunity to work on the validation of this assay when I was working at Illumina. So what the TrueSight Oncology does is that it's a comprehensive panel for genomic profiling, and it uses both DNA and RNA to be able to look at a lot of genes. So from a single test, you can probe several genes and biomarkers. Now, this slide gives you some kind of highlight of what TrueSight Oncology does. That's the name of the test. So you can do genomic profiling, and from one sample, you are able to test for more than 500 cancer genes just from one sample. So I think this test really revolutionized, just the way we think of diagnosis in general, right? Before, it's going to take you probably up to 50 or 20 different tests to be able to achieve this. But with TrueSight Oncology, you are able to do it, right? Have about 550 genes. So it helps consolidate multiple tests, and it also helps inform critical decisions. Then it also conserves biopsy specimens, right? As you know, tissue, we always call it, these are very precious specimens, right? So you can just get, one frozen tissue, and you are able to probe all these genes at the same time. So in terms of the workflow for TrueSight Oncology, what we do in the lab is that, so just say you have a patient that the doctor is suspecting that has cancer. So you collect sample from that patient. So when that sample comes to the laboratory, the advantage with TrueSight Oncology is that you are able to isolate both DNA and RNA. So you have other laboratory tests that will only focus on DNA, and some that will only focus on RNA. But here you are able to probe both DNA and RNA. And the advantage of probing both DNA and RNA, I've listed them here, right? From DNA, you are able to look at your incisions and your deletions within that gene. From the DNA, you can also look at your SNV, those are your single nucleotide variants. And you can also get a biomarker from DNA, which is called tumor mutation burden. And from the RNA, you can look at other things from the RNA. So this could be fusion, this could be splice variants, right? So you don't need to do separate tests to be able to do that. But the challenge in this workflow actually comes at this step, right? Those that have done this, you imagine that doing phase separation is never easy, right? To be able to achieve the target of having both DNA and RNA in the same sample. Then from there, you do your library preparation. Then from there, you do your sequencing. Then you are able now to have a report. So I've just put some information here about splice variants and the fusion genes, what they do and all. Like when you get the slides, you can get into those. So this assay is a pan-cancer panel. As you can see, it covers almost every cancer that you can think of. And here, we've only listed the actionable genes. So these are genes that if a doctor sees it, the doctor can actually take action. So it begins from breast cancer, colorectal, bone, lungs. It covers almost everything. Again, this panel has a total of 550 genes. So what is listed here is just those genes, right, that are actionable. So you see that just from one sample, you are able to probe all of this, right, from one tissue sample. And it's not only probing genes, you are also able to look at biomarkers. And when we talk of biomarkers, the two that I really want to highlight here that is covered in this panel is what is called, the tumor mutation burden and also the microsatellite instability. So tumor mutation burden actually quantifies the number of mutation that is in that a tumor can generate, and it helps predict, if that person can respond to immunotherapy or not. And when we talk of microsatellite instability, it really detects instability in short DNA sequences, right, which can be associated with the progression of cancer or the responsiveness to immune checkpoints inhibitors. So just taking both of them, this cartoon kind of really gives us what is happening in different immune microenvironment. So I'm just going to try to explain it here in terms of how can you use just biomarkers by itself in the lab as a companion diagnosis in terms of treatment options. So you have MSI tumor that is high. So this is microsatellite instability. Then you also have one that is stable. Now, what happens is that during DNA replication, there are errors that occur during DNA replication. And some of those errors are what is referred to as mismatches, right? So these mismatches occur. And when these mismatches occur, your system needs to kick in to be able to repair this mismatch. So in this case, it repairs it very well, right? So you have low mutation that occurs. And when there's low mutation, T cells are not deployed a lot, right, to come fight, that particular cancer. But in the case where this checkpoint is deficient, so you have all these mismatches, it's going to create what is called neoantigens. And when those neoantigens are created, your immune system is going to deploy, right, an army of T cells to be able to fight this cancer cell. So those that have very high microsatellite instability are more likely to respond to certain cancer medication. And also mind you that as the immune system is trying to fight these cancer cells, the cancer cells also are trying to counteract that effect, right, by producing PD-L1. So that's why you always hear of PD-L1 inhibitors, right? That's what they actually do, because you really want this to go through, right? But then the cancer cells is also producing this inhibition, right, to help keep itself. So you need to bring in some kind of inhibition here as well to prevent, this binding from taking place and have that tumor cells cleared as well. So one of the advantages too of the assay, the TrueSight oncology that I've been referring to here, is that it has that ability for you to also do what is called liquid biopsy. Before I left Illumina, we started validating the liquid biopsy version of this assay. So liquid biopsy means that instead of collecting tissue from that patient, we draw blood from that patient. And drawing blood from that patient, you are able to extract what is called circulating tumor DNA. So what do we mean by circulating tumor DNA? If you see here, this is a tumor. As you can see, that tumor is able to secrete or produce proteins. There's also DNA that comes from that tumor, and that DNA is circulating freely in the blood. So there's a special extraction kit that we use in the laboratory, and we are able to extract just this circulating tumor DNA. So you could have a tumor, maybe at the level of the lungs or maybe at the level of the pancreas and all those things, and you just draw blood sample and you are able to still do these same tests, right, using tissue, right, in this case, not using blood. And the advantage of using blood is that instead of tissue, right, tissue, you are focused on a particular location, but blood is systemic, right? So you are able to probe circulating tumor DNA that is coming from several parts of the body, right? That's one of the advantages of liquid of biopsy, apart from the fact that it is less invasive as compared to collecting tissue samples from the patient. So I just want to compare the way we've been doing things traditionally in the lab in terms of companion diagnosis, and how an assay like the TrueSight oncology has come to change things a little bit. So if you look at the TrueSight Oncology Comprehensive, so it provides more information from less sample and in a very, very minimal time. So here you have the TrueSight Oncology. So just take, for example, this is a classic, NSC LC. What is going to happen is that you're going to need like 5 slides if you are looking at TrueSight Oncology. And you are going to probe 517 biomarkers and genes, and you're going to use 5 days. Now, the traditional way that we've been doing things in the laboratory, just say you're going to do like EGFR by PCR, that you're going to use 5 slides. Then from there, you're going to do FISH. So you're going to use different laboratory techniques. So it's going to take you about 42 days to be able to do all this testing. And you're going to look at about 9 different biomarkers, and you're going to go through about 29 different slides. So that is why comprehensive, profiling is really changing the game in the way we look at, cancer diagnosis and companion diagnosis in general. But again, it's not a one-size-fits-all. It doesn't mean that every patient that comes needs to get a comprehensive profiling. No, there are some patients that need just probably the EGFR, PCR. Again, that is mostly the decision, for the oncologist to actually make. Then this is how a report is going to look like, right, for this test. So you do your testing, and you can imagine looking at 550 different cancer genes. You have a lot of data that is generated just from one patient. How do you take that data and make sense out of it is usually the million-dollar question. But the good thing is that now with bioinformatics, AI, and a lot of softwares, it is now possible. So this is how a typical report is going to look like for a patient after we run this test. So you're going to have the patient's information, the name, and everything. Again, this is a dummy patient, so there's really no PHI. They are going to companion diagnosis result, right? Because companion diagnosis, as we said, it has to accompany something, right? So you have the diagnostic test and an accompanying drug that goes with it. So here you see that they were able to identify 2 fusions. And for these 2 fusions, they were able to identify the therapy that is going to be indicated for this fusion that was identified for this particular patient, meaning that this particular cancer patient that has this fusion, if you give this patient this therapy, the patient will most likely respond to that particular therapy. Now, just to put it in a simple way, just say maybe urinary tract infection could be an example that we'll use, right? A patient comes with urinary tract infection, we go to the lab, we do culture, then we do sensitivity, right? So doctors are very familiar with antimicrobial sensitivity testing, right? Where you know which particular antibiotic is going to kill that bacteria the most, and that is what is prescribed to that patient. So in a nutshell, this is some kind of what is happening here. So there you move now to the next section, where it gives the cancer mutation with evidence of clinical significance, right? So it gives the particular mutation, then it gives the evidence, and it cites literature, right, to actually help the clinician. Then it also gives cancer mutations with potential clinical significance, right? So there are some that has clinical significance, there are some that has potential clinical significance. So it's going to list those mutations here that have potential clinical significance. Then, of course, it's going to talk about the quality control, because again, running a very complex test like this, you need to make sure that you're on top of your quality control. Then it's also going to give the companion diagnostic intended use and evaluation. In the case here, it was for solid tumor and also non-small cell lung cancer, right? So it's going to give that intended use for you. So it gives you all this comprehensive information that you, the oncologist, you are able to sit in your office and take all this data now, and it actually helps you in terms of which treatment to give for that particular patient. And it doesn't end there. It still goes further to give you, intended use of the assay and gives you some kind of a summary. And of course, you always have some disclaimers here and some kind of definition of some terms there as well. But most importantly, I think what has really helped is what we refer to as the knowledge base, right? So advanced bioinformatic tools. So gone are the days where we used to do all these things manually, right? You can imagine having gigabytes of data to be able to do all this analysis. So we're able to validate a pipeline. I took part in this validation, which was, I think it took us almost 9 months to be able to get this test ready, right, for clinical use when I was there. So just validating this pipeline on its own, I think took almost 2 to 3 months, right, to be sure that, hey, you run that pipeline, it takes all that genetic information and is able to produce a report that makes sense for the clinician. So personalized medicine is not only for oncology, right? I just want to switch a little bit on infectious disease, and I have I think just 1 or 2 slides on that. So companion diagnostics are also critical infectious disease management, right? Particularly in the area of antibiotic resistant testing. So these tests, they detect genetic markers of resistance in pathogens such as methicillin resistant Staph aureus, multi-drug resistant tuberculosis. For example, I know a lot of people are familiar with the GeneXpert, MTB, RIFF assay. So that's a typical example. So it's an assay that rapidly identifies Mycobacterium tuberculosis, but it doesn't end there. It also identifies its resistance, right, to rifampicin. So that could be a more common example, right, of a companion diagnosis, right? Because you are testing, you are looking at genes, and you are actually saying if a patient is resistant to that particular antibiotic or not. And in viral genotyping, companion diagnostics are essential for identifying mutations in viruses, such as HIV, that confer resistance to specific antiviral drugs. So this ensures that the patient actually receives the most effective treatment. And talking about genotypic resistant testing in the lab, we refer to it as GARD. It plays a critical role in HIV management by detecting mutations in key genes. And those key genes are like the reverse transcriptase, you have the protease, and you have the integrase that confer resistance to antiretroviral drugs. So the process involves PCRO. That's what we're going to do in the lab. So we're going to do PCRO of the genetic region. Then it's going to be followed by sequencing. So GARD is most often used when a patient experiences some kind of a spike in the viral load, or during treatment failure, or to guide clinicians in selecting the drugs that remain effective. So, for example, we have a test. It is called virus-seq HIV-1 genotyping. There are also similar assays. There are some lab-developed tests that are available as well that can be used. So this genotyping actually helps identify these mutations and enabling personalized therapy, thereby preventing further treatment failures and limiting the transmission of resistant HIV strains. So this resistant testing is particularly recommended for newly diagnosed patients and for those that are experiencing treatment failure, or when switching therapy to ensure that the new medication is going to work. So importance of drug-resistant testing in HIV management. Again, as I mentioned previously, it kind of personalized the antiretroviral therapy. It kind of prevents treatment failure and prevents transmission of the resistant virus. And when is the HIV-1 drug-resistant testing recommended? Again, as I said previously, during the initial diagnosis, this will be for new patients, or you have treatment failure, and when you are switching therapy. So it actually helps determine which one is going to be the best for you to use. So rounding up some of the advantages. So I think you've realized that companion diagnostics, they are essential for delivering personalized medicine by guiding targeted therapy. So companion diagnosis, they identify patients, who are most likely to respond to specific drugs, ensuring that the treatment is tailored to the individual's genetic profile or disease characteristics. So this leads to improved patient outcomes, increasing treatment success rate, and most especially enhancing quality of life. So companion diagnosis also improve safety and efficacy of treatment by reducing adverse effects, as only patients who are likely to benefit receive the therapy. And it also further helps determine the optimal dosage, making treatment safer and more effective. And additionally, companion diagnosis, they offer a cost-effective, right? They are cost-effective by avoiding ineffective treatments and minimizing the trial and error approach that I mentioned before. So this direct path, this is the direct path to the right therapy, not only reduce unnecessary treatment, also saves healthcare costs as well. So companion diagnosis, they also play a vital role in accelerating drug development. That's why we have a lot of pharmaceutical companies that are also incorporating companion diagnosis in their clinical trials. So by enabling more targeted patient recruitment for clinical trials, companion diagnosis can actually help streamline the process, increasing trial success rate and reducing costs. So this actually allows pharmaceutical companies to bring targeted therapies to the market more quickly. Additionally, companion diagnosis can aid in regulatory approvals by demonstrating that a specific drug is effective in a well-defined population. Those that have gone through FDA submission and all the like, I know Luis is on the call, you better understand this when it comes in terms of approval. So in the realms of immunotherapy, companion diagnosis actually enhances patient selection by detecting, biomarkers like PD-L1, which I mentioned before, or microsatellite instability. So all these biomarkers, they actually guide the use of immunocheckpoints inhibitors in cancer treatment, ensuring that the therapies are directed to the patient that are most likely to benefit from it. Of course, while companion diagnostics offer significant benefits, they come with limitations as well. Many companion diagnostic tests are developed for use with a single drug, right? Meaning that they focus on identifying one specific biomarker for one treatment. So this narrow scope may miss other treatment options that could be beneficial for the patient. So additionally, companion diagnosis is only applicable for patient that has the targeted biomarker, which limits its use for certain individuals. So there are also cost and access issues. So developing and validating and manufacturing companion diagnosis is very, very expensive. So just doing clinical validation for the TrueSight oncology that I talked about took us about almost 9 to 10 months just to do the validation. And that was just the manual validation, right? Then from there, we had to go now to do the automated validation, right? Where you are able to use Hamiltons and all those things. So give and take about 15 months, right? Just to do the clinical validation. Clinical validation is different from development, right? So developing the test all on its own, that's a different pipeline, which takes an extensive period of time as well. So it's called, right, to be able to develop this test. And finally, the regulatory pathway for companion diagnosis is complex, right? So imagine that you have your test that you are just testing for COVID, right? If you are submitting to FDA, it's direct, right? You are testing for COVID. But now you are bringing this test where you're saying that, hey, I'm testing for this particular mutation, and I'm trying to see if this patient will be able to respond to this drug. So it's like a combo package, right? So it really delays approvals for either the test, or so delay approval for either the test or the drug can, just impact the entire pipeline. So those are some of the limitations in terms of companion diagnostics. And again, too, there are some ethical issues as well, limited access in low resource settings of the world, Again, these are expensive assays to be able to implement, especially if you are looking at next generation sequencing. For the PCR part, yes, there are some good PCR assays that can be implemented, which I think, in Africa and in Cameroon, a lot of thermocyclers that we have now, right? It's just now to be able to run the validations for some of these markers. Again, too, it's going to be markers that are most relevant for that population, right? Those gene targets. So there's still that ethical issue of access. And again, as I always said, not all tests are created equally. So there are times that you can have false negative and false positive test results. These are issues that we see with testing. There are some testing that are 100% sensitive and specific, but not all follow that algorithm. So to conclude, the current way that we've been looking at medicine is a one-size-fits-all approach, where you have cancer patients, there could still be HIV patients, it could be, example, colon cancer, and all these patients are given the same therapy, the same dose. What happens is that you're going to have some patients, they're going to have an effect, right? It's going to work for them. For some, it's not going to have no effect. And for others, it may instead have an adverse effect because the rate at which they metabolize the drug could be low. So here is the future of medicine, right? The future is you have this group of patients. What you do is that you can draw blood, you can get a tissue sample from those patients, and this is where companion diagnosis comes in. We are able to look at or to probe the genes of this individual to be able to say that, hey, this particular patient cannot respond to this particular treatment. So this treatment A, patient A will respond to, but patient B will not be able to respond to because, hey, we have seen a resistant gene in patient B. That way, you are now able to tailor the medication, the dosage, to specific groups of people based off on their genetic makeup. And what happens is that everybody has a nice effect. Everybody is treated. The patient is happy. The doctor is happy. And that takes me to the end of my presentation. Thanks for your kind attention.
Wow. Everybody is happy. The patient is happy, the doctor is happy, and Dr. Mono is also very happy that he asked you to do this presentation. Thank you very much. And Dr. Math, I would like to get your opinion about the presentation. J'espère que ça a été.
Merci Elvis. Oui, c'était très, très intéressant. Je remercie le docteur Kenji de nous avoir partagé en fait les différents aspects de la médecine de diagnostic, particulièrement au niveau de ce qu'il y a comme avancée dans ce qui pourrait être transporté en Afrique, notamment le séquenceur portable. Je pense que j'ai pris note, c'est le ONT, donc j'aimerais l'entendre dessus. J'avais quelques questions, effectivement, il a mentionné à la fin que il y avait des problèmes éthiques à appliquer la médecine de précision, donc j'espère qu'on pourra entrer un peu plus dans des détails dans cet aspect-là. Mais merci, excellente présentation à toi, Elvis.
Thank you very much. And for those who joined late, Dr. Math is co-moderating the session with me, and going forward, she will be co-moderating in French. For those who joined late, today's session has been presented by Dr. Kenji Obadia. So thank you once again, Kenji. I'll pass over to our chair, Dr. Mono, for your comments.
Wow. So this takes us back to the roots of where everything starts. You know, the way the future of medicine is very integrated in science. So majority of what we do in the medical world starts at the level of fundamental sciences and also at the level of the lab, where we are able to help streamline processes that would have taken us years. As Kenji said, it took us more than 5 years to sequence the first human genome. And you cannot believe today they are able to do that in the lab in less than 4 hours. Things that are more complex, cancer genes are 100 times more complex than the human genome, the variation. So it tells you how much and where we've gone to. And we hope, my only cry is always this, is we hope that this technology, and with the use of AI, as we spoke last week, can easily be transported into Africa and take us less than 5 years to catch up with what we're doing in the US, in Europe, and also in a lot of places in Asia, especially in Japan and China. So I'm looking forward to also participating in answering questions. And I'll give you an example of, I have several examples. Just yesterday, I was presenting one of my patients in our tumor board. This young guy had lung cancer. And initially we did, I'll just go through, he's going to go through a lot of the things Kenji did. We came in, we did a PCR, and he had no actionable mutation as per lung cancer profile. That was done 2 years ago. So he was started on chemotherapy because he had nothing. We also did something called an MSI high, which he spoke about. Microsatellite instability high was low. Tumor mutation burden was low. And he was started on the general medicine we give for everyone because we had no actionable mutation. Then one of my colleagues who saw him before I joined the practice decided to do a next generation sequencing. We always fight with the institution at times. And in the next generation sequencing, we had an EGFR mutation, which was a tyrosine kinase a domain duplicate. That changed his life. He was placed on a single tablet after having real resistance to chemotherapy and side effects. He was placed on a tablet called Tegriso, which is called osimertinib. He had been on it for 3 years before I joined the practice. Now he's progressing. Just tells you how we go through this. Now he's progressing. And I come around, he's progressing. He has some oligometastatic disease progressing in some places and not. I get radiation oncology involved. They do some radiation there. And we do another next generation sequencing. And I realize he has developed mutation in MET inhibitor. So there is a skipping mutations that he has developed, which is causing this resistance. And I'm able, because of this information, to add a MET inhibitor to his treatment. I just saw him last week. He has a complete response. He's just 36 years old, non-smoker, but with lung cancer. This is the amazing things the lab is transitioning into the field of oncology and other fields. Medicine is moving forward. I can tell you hundreds of this type of cases, which we're going to discuss. And Elvis, this is really what Kenji is talking about. This is how we're making a difference in people's life. I hope we're going to be able to make a difference like this in Africa. And as he said, companion diagnosis is not just about NGS and liquid biopsy and all these complex things. It starts from blood transfusion, as President Obama said. It starts from R2. It starts from basic things that we're able to do and that we're doing right now that we can invest in. So thank you.
Thank you very much for those really great insights, Dr. Muna. And of course, we have Dr. Sydney, who is here. And I'm sure you might have some thoughts around how this applies to the field of radiology and radiotherapy and so on. Sydney, some comments from you? We have Sydney. Okay. Well, I think I'll pass it back to Dr. Matt for the question and answer session. Please, if you have questions, you can raise your finger up, and then Matt is going to call you and moderate this session together with myself. You're mute, Dr. Matt. You're still muted. You have to unmute.
Okay. Oh, sorry about that. I was saying that Franklin Maloba has his hand up, and Dr. Ekiti as well. So Franklin, you can take over.
Yeah, thank you very much, Madam the moderator. And thank you very much, Dr. Kenji, for this excellent presentation. Sincerely, I've learned something that I don't know where I would have learned it without a town hall, sincerely. Yeah, and I think that with this said, I think that I'm proud as a scientist that we are moving forward to really change the world with such an initiative and with this type of project. Yeah, I hope that maybe Africa could benefit one day. But with respect to Africa, we are still a bit backward with many things. I don't know, how could Africa benefit? Because, you know, when it concerns genetic stuff, there's a lot of ethical issue, as you mentioned at the end of it. So for African, it shows that we'll still be depending on the Western world for this type of medication. So how would the genetic consideration in terms of the patient genetic information be transitioned to protect our data? You know, like the Indian Americans, so far, they refuse to participate in any genetic study because of what happened to them, you know, where their data were violated. So to you, how do you think this can benefit Africa while protecting their information? That's my first line of the question. Then secondly, you know, there's a lot of epigenetic changes that is always occurring due to nutrition, climate change, environmental factors, lifestyle, things like that. So sometimes are there not mutations that could occur to some of these markers, or are there not new markers that can always come out that can also tend to limit some of this precision medicine? Thank you.
All right. Thank you so much, my brother, Dr. Maloba, for those great questions. The issue of ethics in genetics data is a real problem. It's not only an African problem. So it's a global problem. And the way most of this is happening now that I've seen work is having memorandum of understanding and having data sharing agreements as to what you can share and how that data will be protected. But I think most importantly, having safeguards. I'm sure you've gone through most of those training, you know, as a researcher, how you safeguard data. Even here in the US, we have very strict HIPAA policies, right? Just in terms of patient data, who is supposed to have access to patient data and what you are supposed to do with that patient data. So I think these are discussions that needs to happen before anything has to be implemented, not after. You need to think through the entire workflow as to, hey, when the sample comes into when you get that result, what happens and how are you protecting patient data from there? It's never going to be a one-size-fits-all, and I don't think it's an African problem. And for me, I think one of the best ways of solving things like that is to come out with all those memorandum of understanding and having very good, you know, cyber security firewalls and all those things that can help protect that information. In terms of how this can benefit Africa, I always say, I don't want to die ignorant, right? If I want to die, I want to know that, hey, this is what killed me. Or if I lost somebody, I want to know that, hey, this is what happened to that person. And maybe I can take some actions on my own part. Now, the question has always been, okay, if we deploy, because, you know, I was discussing with my team here, if we deploy a next generation sequencing assay to Cameroon that can look at EGFR, and it already has, you know, we already know that the patient can respond to this or that therapy, and then the people are unable to afford that therapy. Doesn't make any sense. For me, I would say, first of all, if you don't look for something, you will not know if it is there or not. I was just discussing with one of my students yesterday who was saying that now there's a lot of cancer in Cameroon. I'm like, I don't really think there is a lot. I say, I just think it's because now we are looking for it. We are looking for it, and now we are knowing that you have this or you have that. Before, somebody may die, and maybe you are blaming your grandmother in the village, right? Meanwhile, probably was, you know, some cancer that person died from. So I think it starts with diagnosis. If we can advocate and diagnose people to know that, hey, I have this, because my brother Maloma, you look like in the US, when you go to see your doctor, one of the things that always asks you, if do you have any history of cancer in your family? Just knowing that, hey, my grandfather had prostate cancer, it could be that, hey, maybe you are at risk. So you better, you know, take precautions if there are any precautions, or you do follow up. So just with diagnosis alone, I think it's better for us to know. Then we can start advocating, right, for the treatment. Some people could fly out and all those things if they can afford. Now, in the African context, we already have the thermocyclers. From COVID, there were a lot of thermocyclers that were bought. And some of them could just be sitting in the lab and probably nothing is happening there. We must not start with next generation sequencing, right? We can still just start with just regular PCR assays. Then from there, we can continue to build up to our next generation sequencing. And even in terms of next generation sequencing, a lot of African countries now have sequencers, right? I had partnered with one of the organizations. I don't want to call the name here since I'm not talking on their behalf. And we did a lot of training in Africa, and most of the countries have these sequencers. Now, the thing is, we need to broaden our scope of thinking, right? The focus, I think, in Africa has been a lot more on infectious disease, which I get it. Malaria is an issue. It's an acute disease. If you get it, you can die in 3 days, as compared to cancer. That is a little, I can say, a little more forgiving, that, you know, it takes a long time to be able to develop. So I think it just takes people to be able to think a little bit way beyond infectious disease. And I feel like most of these instruments that we currently have that we got from COVID, they are extremely underutilized, because now the sample volume for COVID has gone down. Like, I'm looking at GSAID submissions and all those things. It's like nowhere, like submissions you get in 1 month was like submissions that you used to get in 1 day. So they are extremely underutilized, and we can leverage from there, right, to be able to implement some of these diagnostic tools. And I think your next question was on epigenetic changes, if it is going to change the actionable mutations. I don't know if I got that question very well. I think that is where, so in terms of other mutations, is going to affect PCR more as compared to sequencing, because, you know, with PCR, you have a specific, you know, probe, primers and probe, right? Where at that your target, if that your target mutates, that probe will probably not bind, and you are going to have a false negative result, right? So those are some of the limitations if you are using PCR. But if you are using sequencing, it overrides that limitation, right? Because you are actually looking at the sequence of that genome. And this issue is not unique to oncology. If you look at even COVID, right, we started having different variants, right, that were coming up. You heard about the S dropout, right, because there was a mutation at that particular gene. So those primers were no longer functioning, right? So that's why from time to time, you need to do your blast, right, to make sure that these primers are still working for those particular targets. It's a general term of rule in all my laboratories. I think it's every, I don't know, 1 or 2 years, depending on the pathogen. We take those primers, we blast them just to make sure that they are still sensitive. I hope I did answer your question.
Yeah, thank you very much. Yeah, then the last thing I would just like to know is, how much does it cost to implement such in a developing world? And, you know, with infrastructure, these things in Cameroon, the poverty rate, and to make the cost a bit cheaper for the population, what do you think can be the way forward?
I always give the example of CD4 count. When HIV started, CD4 count was just for the rich. But with a lot of advocacy, my brother Maloba, I'm sure you use the PIMA machine in Cameroon. So now it's now affordable, right? Everybody now can do the CD4. So I think these things come with a lot of advocacy. We need to advocate. We need to be our voice. Whenever I talk with a manufacturer, yesterday I had a call with one of them, right? I'm like, hey, can you do this? What if you are able to develop these primers and probe that allow lyophilize, that you can deploy it, make it cheaper in an African market? But the reality is that the manufacturers and the farmers and all those things, they go behind the money. And truly, the money is here. So telling them to develop something that is more focalized for the developing country is going to take some time. It will be good when you have a group of researchers like, hey, you know what? Let's come together and let's see how we can develop some custom NGS protocols that is suited for the African scenario. One of the projects that we had to do, I think that was in Zimbabwe, we produced lyophilized reagents for the primers and probes and everything. That was very, very good for that particular setting, where when it comes, you just reconstitute it. That will be good there. So I think with advocacy, we can get there. When HIV started, it was the same question that people had, you know, in terms of CD4 count. But with advocacy, we are slowly getting there. And in terms of cost, I would say, I think the overall cost that we had calculated in another forum, I'm just giving the example of COVID, just to do the entire sequencing and everything for one sample, I think was getting about $150 just to sequence one sample, right, for COVID, right? So for oncology, it will be different too, because again, Like here in the US, labor is a big thing, right? So, so you take all those things into consideration too. So I can't really give a figure because it's not something that I've ever implemented in terms of oncology in Africa. Thanks.
Thank you.
Thank you, Franklin. I'm going to take Dr. Kitty Martin.
Thank you, Martin. Thank you, Dr. Kenji, sorry. Well, I always say that it's always great being on the town hall because in an hour you get to learn so much that you probably need a few lectures or courses in the university to learn, and it's always interesting and fascinating to see how we condense such complicated things into such digestible language that any person who is not even conversant with everything in that particular sphere of influence can understand. So now I can definitely say that having no clue from the start, I have a much better understanding now of what not only companion diagnostics is all about, but precision medicine or precision diagnostics or just personalized medicine in general. So my question is more, for me, I mean, I'm a medic, so NGS is probably next level, way over my head. I know Dr. Mono, he is an oncologist dealing with these things on a daily basis. Those things are so far up my, above my pay grade. So I don't want to get into things like that. Let me talk about something that I actually had some experience with, something like IHC, immunohistochemistry. This is something that, for instance, in my time in Cameroon and working with patients who were diagnosed with cancer, and we have labs in Douala that will say they do IHC. And then you send the patient to Douala, they will take a sample, we get the results, and it comes from SEBA Alliance, which is an institution in France. So my question is, to the best of your knowledge, Dr. Kenji, and also any of the other clinical pathologists and, clinical chemists on the call, MLS people in general, what is the regulatory framework in Africa and Cameroon especially? And why? Because when I was in clinical practice, one of the issues that we had with the lab in general is the ability to trust the results that come from a lab. And then you have a situation where the doctor will say, no, don't go to this lab, go to this other lab. And, some of those things have a negative impact on the psychology of a patient when they know that the hospital that they are being consulted, they shouldn't do a test, they should go somewhere else. Now, that's just one of the factors. There are several other factors that we don't want to get into here, why we send people to different places. But what are the factors that that have an impact on the reliability of testing results in Cameroon as a whole? Now, in the US, we have things like, clear accreditation, we have CAP, we have institutions like CAP, like Joint Commission. These are people that go around making sure that labs are doing what they're supposed to be doing and the results are reliable. Do we have such frameworks in Cameroon? And if we don't, I really hope that they are. I just don't know about them because I'm not in that particular field. But if we don't, do we even want to start getting into things like these high next level things that are problems in the developed world, when things like regulatory framework and infrastructure to make sure that there's reliability and accountability of the entities that are involved? So I don't know, to the best of your knowledge, all of our clinical pathologists, clinical chemists, our people in the lab on the call, I just want to be enlightened, I would say. Thank you very much.
Yeah, I think I can go first since I've worked in both settings. I'm not sure if much has changed from when I worked in Cameroon to now, but I have a few of my students. Elad is on the call too. Elad is more familiar now with Cameroon. So in the US, yes, we have CLAIR, we have COLA, we have CAP. We recently just had CAP inspection 2 days ago, right? So you have these people that are behind you. You must do it this way. In Cameroon, I don't think there's any body, right? There's any formal body that directly regulates the laboratory. It could be on paper, right? Where you have all these texts and all the like, but throughout my working in Cameroon, I've never had an inspector come to the lab with a formal kind of checklist to be like, hey, you are compliant or you are not compliant with this particular, checklist item. That notwithstanding, it doesn't mean that all the results from the labs in Cameroon is garbage. No, you have labs in Cameroon that are ISO accredited, There are several labs in Cameroon. And ISO is an international standard, right? It may not be CAP, but it's kind of similar to CAP, right? So at times, a doctor will recommend, like even me, if I'm sending somebody in Yaoundé, I'll say, go to an ISO lab, right? I don't want to call names of labs here, but there are a few that I know in Yaoundé and in Douala that are ISO accredited, meaning that they have inspection. And WHO had also started an accreditation program as well, which I know the regional hospital in Bamenda, I think, went through that program, right? So we do have, I would say things have improved as compared to a long time ago, right? There is some kind of that awareness in terms of quality in Africa in general, right? Before this call, there's another seminar that's taking place in South Africa just on QMS. So you have a lot of these workshops that are taking place now in QMS. But in terms of that kind of US standard regulatory body, it's not there. But I think we are gradually getting there. And again, I always say not every test is created the same. Even in the US, the labs are accredited, but you can still go to a lab and you have a false positive or a false negative result. And it all just boils down to what is the limit of detection of this particular assay. It's the same during COVID. You test here, it's positive. You test there, it is negative, right? So at times too, it could just be limitation in terms of the assay that that particular laboratory is using, not that the lab is not implementing quality measures. Now, in terms of implementing next generation sequencing, Cameroonian scientists are very smart. I train in Cameroon, and I've gone around training in Africa as well. You go to a 2-3 day training. I think Cyrus here, he attended one. 1 week training, they get it. Like they are able to do sequencing, they go to their lab and they're able to implement the sequencing. So in terms of that knowledge and in terms of people grasping that technology, I think we are good. I think where we really need to shift now to is like, hey, how do we formalize a body? I know in Cameroon, we have CAMELS. We have another association for labs. How can we have that independent body that could be like, quote unquote, I know regulators don't want us to call them police, but quote unquote police, right, for the lab. Before you open up a laboratory, these are the things that you are supposed to meet. You are supposed to have, like in the US, you cannot open a lab if you don't have a medical director. If it's a high complexity lab, the person must have at least a PhD or an MD, PhD, with residency in clinical pathology, right? You cannot open a lab if you don't show the credentials of that person. And when you are board certified, you can only manage 5 laboratories, right, as a medical laboratory director. So before Claire gives that approval, there is that director. You must also have your technical supervisor. You must have testing personnel who also needs to go through an accreditation process. They need to write the ASCP exam and pass the ASCP exam. But you see that in most, not only Cameroon, in a lot of African countries, once we left UB, we just ship directly to the lab, right? We are running whatever we are running. The fact that you went to school doesn't necessarily mean that school went through you in terms of you being ready to work on patients. That's why even medical doctors, they still need to write all those examinations. Even here, you need to take boards upon boards upon boards before they are like, hey, because dealing with humans is very critical. We need to make sure that whoever is dealing with humans is well vetted. So it's from several fronts, right? Accrediting the lab and also accrediting the personnel. But I would like to get a viewpoint from those that are currently in Cameroon, Ella and the rest, if there's anything they want to chime in.
I think I work in Cameroon and I can throw a bit light on that. Yeah, one thing that brings these issues too is the fact that in Cameroon, we rely on a lot of, district or maybe integrated health center hospitals. And some of these hospitals too don't have the appropriate facility to store most of these reagents to the appropriate temperature. So at certain points, even most of these reagents are not more productive to the way they can give the best result. And that is going back to the lab technician who may be doing the right job, Like, for instance, you go to some health center, they stay for 1 week without light. Have they store all their reagents within this time? And they bought this reagent. What would they do? Would they throw them? They will manage to use those reagents to the best way that they can use it. And sometimes this reagent cannot give the appropriate result. Second thing that I've noticed sometimes too, I don't know if it's administrative bottleneck in the health in the health facilities. There are many medical doctors that once they look a patient, they give a diagnostics. Sometimes this diagnostic may not even match to what the patient is complaining about, but there's no communication between the lab and even sometimes the director. He just writes his paper, go and do this test. The person in the lab do the test and send it there, and there's no counter communication to see, but what is overall in this patient? What do you think we can do next for this patient that is negative? You know, we just say, oh, your test is straight negative, you are good to go, you are not sick. You know, we just limit ourselves to, there's a lot of limitation, and this is due to the number of patients that a doctor faces, and also the number of patients that a medical lab faces. You go to lab, like for instance, when I was working in the north, we are just about 5, we have 200 patients a day running liver function, kidney function. The workload is too much to even think otherwise around what was given to you. The doctor too, you can't blame them. There's one doctor is to about 1,000 patients. How will you blame them? So sometimes it's just due to the fact that the system needs to be fixed, and also accreditation body, as Dr. Kenji mentioned, that can always try to make sure that these scientists are upgraded to the new diagnostic methods that are available. And also availability of the new diagnostic methods is an issue. We are still most in the archaic method, like typhoid diagnostics. We still use Bewida. You know, people still use Bewida to diagnose for typhoid, and we should know that Bewida is not the real best test. But what do? We just go with what is available. But now people will put it like maybe it's a lab technician issue, but meanwhile it could not be. So I think that is just a lot that we can write a very big book that will not end this today. Thank you.
Okay, so I will put a little bit, I'll give a little bit of an input. I trained in Cameroon as a clinical pathologist, meaning exam biologist, and all what Kenji said was really the fundamentals. How do we, in the first place, accredit our labs internally? As Kenji will tell you, there is internal control in the first place that has to be taking place by the lab director, and there is external control. So when I was in CSU, I think then in all the different labs, I'm not going to call names, but I think the people will be happy for me to say, I worked with Professor Nkonga in biochemistry. We did internal controls of our reagents. And others who are out there may not know. And we discarded reagents when we discovered that they did not meet the standards. And we had the ISO accreditations. At the time, we're looking for accreditation bodies that would send us samples. I'm not trying to guide, people need to know that in Cameroon, people are trying to do a lot. Even in the Shanta Biya Foundation, Fanta Biya's research center, I've forgotten how it used to be called, Professor Tori Miro and the others, those were all things they did in order to test their primers before using them when they're doing HIV testing and resistance. In Centre Pasteur, they did the same because they were accredited. All the Centre Pasteurs actually work with France. So there is a lot being done. I know in Bamenda too, the WHO accreditation process is taking place in their labs, and they're doing a lot. Now, when it comes to certain tests, is the way we train our pathologists, the way we train our medical scientists, and the way we train our physicians. Now, how do you get into a point of testing IHC if you have not made people understand that it's important? So it's not really an aspect that we could not do it. It's actually still an aspect that we don't know the importance of doing an IHC. So IHC has a lot of other problems when you start doing them, but I think we don't even request for it in the first place. We just started requesting for it when advocacy came into breast cancer. Nephrologists need it a lot in order to decide what kind of chronic kidney disease you have. It's actually very relevant in nephrology and nephropathology. How many pathologists are trained in nephropathology? How many pathologists are oncology pathologists? How many of them even feel the necessity that, okay, this woman has breast cancer, let's do ER, PR, and HER2 stents. If you don't, now that we're requesting in bulk, even Centre Pasteur could not do it in Cameroon because the demand was low. So they sent it to Senegal in order to put those things together so they don't. You have to be careful with IHC. IHC has a problem with subjective interpretation if they don't do it well. Even in the US, not all labs do IHC. Trust me, HER2 interpretation, Kenji is going to tell you, HER2 interpretation is the most controversial interpretation in medicine. You may not even know that, but it's a difficult interpretation. It's easy to say your HER2 0 or your HER2 3+. In between, nobody knows. Nobody really knows because we have taken 20 pathologists and put them in a room, 100 of them, and for them to differentiate between HER2 1+, 2+, and the results were awful. Quality and consistency of antibodies that we get is a problem. Also, technical variability varies depending on the things that you get. There is also limitation in the qualitative precision methods that we use. Cross-reaction and non-specificity in binders that we're using for IHC. So IHC in the first place, we need to, I don't think, Ekiti, this is a big area where we can invest in Cameroon, as much as we would have the samples to do it, and we'll have physicians requesting for it. And I think big hospitals like La Cantine in Douala, where people have money, they are supposed to be able to do that. L'Hôpital Général are supposed to be able to do that and pool their forces together. They have all the specialists that have to be able to do that. Then the laboratory medicine in Cameroon is full of very intelligent young men. For them to learn and do IC and do it consistently will not be a problem. That's my own take about it, because I did work in the lab a little bit in Cameroon, and I can tell you the innovation is very, very high end. Cameroonians adapt a lot. There's nothing, they used to do drug testing for HIV like nothing in Cameroon, like nothing you can imagine. And they're able to tell you, but the physicians even request for those results, they don't want to know. And what do we do about it? So training, recertification, getting programs in, getting a licensing body that is going to help us accredit our labs at different levels. Every lab at every level has to have a test that is able to do. My hospital I used to work in, SEMA in Mancon should not be able to do certain tests because they don't have the capacity to do it. District hospitals should be able to have a limitation of what they can do. And pool the forces so that the regional hospital in Bamenda is going to be able to do some of those tests and send to them immediately. Then you have other central labs should be able to pull it. Cameroon, not everybody should do an IHC, but we should have a central system which is efficient, that we don't send it out and they can do it in 1 or 2 days and send the results worldwide. Even in the US, not all pathologists do certain tests. They stain test and they send some out. Even PDL1, you know, Kenji, you guys are a big force where a lot of results come in and you guys send them out to the whole Standford system. So not all labs, even in the Standford system, are able to do certain tests. So thank you. That's just my little input.
Thank you all so much for those contributions. They have been enlightening. There's a lot that we can do. There's a lot that, in fact, even before a lot that we can do, there's a lot that we are already doing. There's a lot that we're already doing. I will need to optimize some of the resources that we have to use them to better effect. Another thing is communication. I've noticed here in the US, there is not 100% great communication between the pathology department and the clinical specialties, especially it's not across the board, but in certain areas, you know that there's some communication that is wanting. But I don't know, like when the nephrologists have their because of the Cameroon Society of Nephrology, when they have their meetings, you know, as pathologists there to talk about pathology, if it is something that is, you know, let them know what's important. IHC is important for nephrology, for instance. I'm just throwing things out there, but there's some low-hanging fruit, I believe, you know, without reinventing the wheel, without breaking the bank. I think that there are ways that we can all work together to improve communication and just optimize the resources that we already have and the know-how that we're already very good at assimilating, like you all have said. Thank you all very much.
Warren, we can go ahead. Warren Cyrus.
Yeah, good afternoon from my own end, and thank you very much, everybody, for this such an educative session. Dr. Kenji, I have learned so much from the insights and also the concepts that you shared with us regarding the aspect of companion medicine, of which you made mention that metabolism can be one of an important element. So you rightly said that in cases wherein you, depending on the scenario that you created around the patients, how they respond to certain treatments and certain drugs, like intermediate, minimal, and other cases, you made mention that there are certain patients who has low metabolism rate, normally respond slowly. So I wanted you to like unbundle the concept behind this metabolism, which is one of the things that I'm interested to know, because I am a lab professional. So I really want to get a concept behind the aspect of this word metabolism. And again, you also made mention of microsatellite instability, which I believe can be also another important thing that I need to know. I need to wrap my hands around, because you made mention that in certain cases, while doing some diagnosis for certain patients, there are some that can have low microsatellite instability and others that can also have low microsatellite instability. And more or less, moreover, they can also affect the rate at which the concepts of this mismatch repair normally occurs in certain patients. So I wanted you to make some clarification around that. And thank you very much in advance as I wait to listen.
All right, thank you so much, Warren, for your questions. I will start with microsatellite instability, since that is what I'm more familiar with. So the way you can kind of think of microsatellite instability is when you have DNA replication, there's always mismatch basis that is going to occur, right? Just calibrate your head back to COVID, right? It's an RNA virus. During replication, there's a lot of errors that are introduced. That's why you have all these mutations that come about. But for DNA replication, you are supposed to have a mismatch repair mechanism where when you have those mismatch bases, it kind of corrects it, right? So once it corrects it, you say it is stable, because we talk about microsatellite. These are repetitive regions in the DNA, right? That's what it means. Those are microsatellites. And those are those regions that are prone to all this mismatch. Now, you have a scenario where it is stable. That's the first one I've given you. The second scenario is when that repair mechanism is having an issue in a cancer patient. So that mismatch doesn't get repaired. So what happens is that this person accumulates a lot of mutation and produces what is called neoantigens. Now, these neoantigens now is going to prompt your immune system, and in this case, they are the T cells. It's going to prompt now the T cells to come and fight that tumor. So it means that somebody that has microsatellite high that is unstable is more likely to respond, right, to your immune system and to respond to certain therapies as well. But also mind you that as your immune system is trying to respond to that tumor, the tumor also wants to survive. That's where the PD-L1 receptor now comes in. Because once it engages it now triggers an anti-tumor mechanism. So that is where now Dr. Foma will come in and like, we need to give this person a PD-L1 blocker, which is going to block that binding and allows your system to be able to fight that tumor. So that is it about microsatellite instability. So for somebody because as we are sitting, replication is taking place, mismatch is going on. A few mismatches will not really trigger that many T cells to come, kill that tumor. So that's why somebody with high tumor mutation burden and unstable and microsatellite instability high is more likely to respond to specific chemotherapy. So in terms of drug metabolism, I mean, that is where a pharmacologist will come in place, to give you more. I won't say it's so much my specialty, but I already told you about SNPs. So these are polymorphisms, right? So these polymorphisms, depending on the kind of polymorphism that person has, the person may not be able to respond to certain drugs. And again, for drugs to be metabolized, you need enzymes, right? So you can still have deficiency in these enzymes or polymorphism in some of these enzymes, right? That make that person not able to metabolize that drug. You can also have enzyme inhibition, right? Of course, if you go like to antibiotics, right? So you have some enzymes that can inhibit, certain drugs. So for example, taking a drug, that inhibits CYP, at least CYP3/4A alongside. Sorry, you can also have what is called drug-to-drug interaction as well, right? Where you have those competitions. So it's a lot of things that come together to be able to make somebody a slow, metabolite or a fast metabolite. It's a complex set of mechanisms, which I don't think I can give you a very, very comprehensive response. Probably if we have a pharmacist or a pharmacologist on the call, the person will be able to give you a better response than what I've given.
Let me just show you something. Okay?
Just gonna add, I don't know, Dr. Mono.
You listen to, yes, please. Let me just show you something which is important, and I'll add to everything that Kenji has also said, which is what we do. So MSI high lesions are occurring all the time, and the immune system senses that, and your T cells want to go there and fight. So you need to understand that fighting cancer is something your body does every single day. Your body fights cancer every single day. You can't believe it, but you make so many cancer cells that your body's job is to fight and kill them. And how does the body know? The body has something called MHC class 1 and 2 that they express antigens. As much as your genes are changing and microsatellite is not stable, the body is actually expressing that. And the immune system is moving towards that. I don't want to go into the detail. MHC class 1 and 2. Now, the cancer, as Dr. Kenji said, which I'm going to show you here, decides and says, okay, sends out PDL1 means program dead ligand. It sends out a signal saying that do not kill me. The immune system is super interesting. And when it sends out the system saying that do not kill me, your T cells come there and they engage with it. They are married to it. They say, okay, this is a good guy. Don't kill him. And they go away. That's how cancers escape death from your immune system. What you should know is that your immune system surveys all your cells daily, and it catches cancer cells and kills them every single day. So this is how we decide. There are multiple ways we decide on whom to get immunotherapy in the first place. Everyone who is MSI high responds very well to immunotherapy. Those who have a high tumor mutation burden, meaning that their tumors are mutating, and there's another way of us picking it that Kenji described very well, they will respond to immunotherapy. Those who have a high PDL1 expression will also respond to immunotherapy. So this is a short 2-minute video.
In recent years, immune checkpoint blockade has taken center stage as a significant breakthrough in medical cancer therapy. Activation of T cells involves several paired molecular interactions, such as T cell receptors with the major histocompatibility complex, B7-1 with CD28, and several others.
I think
Programmed cell death receptor 1, or PD1, is a member of the CD28 superfamily and is expressed on T lymphocytes.
Yeah, the video is kind of breaking. I don't know if it's just from my end.
In recent years,
same here.
Yeah, Dr. Fomati,
immune checkpoint blockade
we may not be able to get the animation. It's breaking.
Yeah, this video is breaking. Sorry, guys. Were you getting
breakthrough in medical cancer therapy?
Exactly what we just described, and I'll be able to share this with some of you here, just to understand the issue of the major histocompatibility complex, PDL1, and that's where we have created antibodies, which are called PDL1 inhibitors and PD1 inhibitors, and that's actually one of the ways we do immunotherapy in oncology. So I will not, let me share something. I'm not going to share a video. Let me just show you exactly how that works in terms of the slides. It's going to be short and brief. So here you can see, let me see here if I can go back a little bit. Okay, yeah. So as Kenji will tell you, these are the things, these are the innovations that are being worked on in oncology right now and in other fields. These are mostly done by those in the basic sciences, not physicians. All these are the interactions that actually occur at the level of the cell levels. PDL1 is just one of them that we are able to block now We do have, they're working on drugs for all of these different markers that you see here and many more. Team 3, there is actually a drug in the pipeline. There's LAC inhibitors that just had a drug in myeloma and is going into lung cancer and other cancers. Here, this is a LAC inhibitor, relactinib, which is a new one, but PD-L1s have in cancer care. So I would stop here and I would try to share this video with some of you who are interested, okay?
Yeah, Dr. Mono, I just wish to step a bit for the metabolite stuff. I just want to use a simple scenario as we are Cameroonian. When Dr. Kenji was talking about metabolite, man, Sige, I know you drink a bit because I usually drink export when I was back in Cameroon. You know, there are some people that can drink 5 export without getting drunk. There are some people that can drink just 1 export, they get drunk, you know. So it means that those who drink 5 export, their system easily metabolize alcohol. That's why they can take more alcohol without being okay. But there are others that, you know, their system, they don't have the enzyme to break alcohol very fast. And when they take 1 alcohol, they are just gone. So I hope he makes it clear. Dr. Kenji, I don't know if you correct me. Yeah.
Yeah, yes, and I forgot about that part of the question. I'll give you a clear example how we use it. So in oncology, we do prescribe patients who have colon cancer. We give them a regimen which is made up of 5-fluorouracil. And also some of them do take oxaliplatin. We do know we can send samples to Kenji in the lab and he's able to study and tell us that these patients are high metabolites or low metabolites. These patients, usually we have realized that those who do not metabolize 5-fluorouracil or capecitabine tend to have drastic side effects and can actually die from the first treatment we're giving them. It's a very rare gene, but there are populations where it's very common. In areas where it's common, it's important for you to test for it. There are also monoclonal antibodies like cetuximab, where in the US, those who live in the East Coast tend to have more side effects because of the interaction with antigens that are occurring also with babesiosis, with ticks viruses and other things, and bedbugs and other things that is making them have more susceptibility to reacting to these drugs. So this is a concept. Metabolism is a primary concept. As he said, we don't metabolize drugs equally. Those who have done pharmacodynamics and pharmacokinetics will be able to tell us more, but I'm just giving you a practical example. Each time I put someone on 4 forks and I'm suspicious, I send for a specific test, and the lab, Kenji works, they're going to send me a result and tell me, oh, this guy is a normal metabolite. I know 99% of the time that's what he's going to tell me. I had one patient about 5 days ago, and it saved him from having the first dose that was going to cause him severe diarrhea and other things. So that is how really this works. And it's a true field where we are still exploiting, and we hope we get more tests that will be able to help us generally, like what we have with NGS and other things.
Thank you, Dr. Mono. I don't know if I have the chance to ask. I just have 2 questions, and I should pass the mic after to Dr. Toignou. So thank you for the insightful talk. Je vais poser les questions en français, si ça ne dérange pas, mais je pourrais switcher en anglais pour faire plus de précision. Donc, il y a 2 questions que j'ai. La première, c'est pour faire suite au commentaire qui a été fait tout à l'heure par rapport au métabolisme. Quand on parle de médecine de précision, on n'a pas le choix aussi de penser à la catégorisation des groupes ethniques. Parce que quand on identifie en fait des marqueurs, souvent ils sont associés à certains groupes ethniques. Alors la première question que j'ai, c'est comment est-ce que vous mitiguez le risque en fait de stigmatiser certains groupes ethniques avec les résultats de séquençage ou même des autres résultats de médecine de précision. Je pose la question parce qu'il y a quelqu'un qui a mentionné que le séquençage est quelque chose qui est plus élevé chez les médecins qui ont une pratique plus simple, qui ne sont pas des oncologues. Alors, ils vont souvent avoir tendance à placer les marqueurs par catégorie ethnique. Alors, j'aimerais savoir comment vous, lorsque vous donnez ces résultats-là, vous envoyez, est-ce qu'il y a un avis pour préciser effectivement que il faut l'interpréter par rapport à la race. Quel est le processus de mitigation, en fait, de l'interprétation? Ça, c'est la première question. Et la deuxième question, c'est plus une question philosophique un peu, toujours dans le partage de résultats. Lorsque vous faites de la médecine de précision, vous utilisez des études cliniques qui ne sont peut-être pas représentatives, en fait, de tous les groupes ethniques. Alors, comment est-ce que vous, j'essaye de mieux formuler ma question, comment est-ce que vous vous assurez que le prescripteur qui reçoit ces données-là l'applique dans son contexte? Parce que ces marqueurs, en fait, sont générés d'études qui n'ont peut-être pas toute la représentation des populations diverses, africaines ou bien latino, mais qui sont plus faites dans une population caucasienne. Comment est-ce que vous vous assurez que le prescripteur l'interprète dans son contexte ethnique pour ne pas causer d'effets indésirables chez le patient? Ce sont mes deux questions.
Yeah, now I regret why I failed French in from 5 and also in the university, because Elvis usually tells me that I mother French. So I'm going to try to see if
I can actually summarize. I think the first question is more around, we know that when we talk about diagnostic medicine, we tend to actually, at least the general physicians tend to group it as race, around prescription based on race. So how do you actually make sure that the prescription, the prescriptors or the healthcare professional in this case is actually differentiating between what is being, the result that is sent to him and the patient that is treating, so that it doesn't cause any harm? So that's the first question. And I can repeat if it's not clear.
Can I answer the first one, then we'll go to the second? So the first one, I'm understanding that you're trying to ask a prescription based on race, how we make sure that all of that is kind of incorporated. For me, I'm not sure if there are specific drugs that you have to give to people because of their race to begin with. What I understand is that, especially for sequencing, it doesn't matter whether you are a Cameroonian or you are from America. The good thing with sequencing is that it is sequencing your own genome. And that is where personalized medicine comes in place. So it's not a one-size-fits-all. Now, that is why sequencing your own genome is going to tell us that this particular medication can work for you, right? It's not that this particular medication will work for Cameroonians. It's working for you. And again, you coming from Cameroon, it could be that you have some polymorphisms that is peculiar to Cameroon. So you may see that if you have people from the US and people from Cameroon, maybe people from Cameroon may respond better to a particular class of medication just because of this particular type of polymorphism that is found in that area. But I'm not sure, but Dr. Fuma can correct me, I'm not sure that in terms of prescription, you want to prescribe a particular medication because it is for this particular race of individual. But what I understand is that there are certain diseases that are peculiar to certain races. So you can talk of things like thalassemia, right? That is peculiar to, certain groups of people. You can talk of Blacks, being more predisposed to certain kind of diseases, right? Yeah, but in terms of race and genomics, we are sequencing your genome. And when we run it through the pipeline will be able to decipher or select which particular resistant markers you have. And there we can, the doctor can prescribe based upon that.
Thank you. Yeah, I think the question was more, go ahead.
Yeah, no, just to add is, we try as much as possible, Dr. Matsandrin, you can see that patients will come to you. And the good thing about NGS sequencing, as Dr. Kenji said, is that we're picking out mutations that are found in you as an individual. We're not talking about the population. Some of those mutations could be associated to certain ethnic groups, which is right. But at that case, we're dealing with you. I'll take, for example, those who have EGFR mutation, which we've talked about in Kenji's case presentation. It's more common in Asians than we see in Caucasians. If you go to China and you do an EGFR for those who have lung cancer, almost about 30 to 40% of them will be positive. In the US, it's less than 15%. But once we see it in you, no matter your ethnic group, we treat you for having EGFR. Then we get a counselor. Let's take for BRCA. BRCA is more common among Ashkenazi Jews. That's a fact. We're also starting to see it among Black people because we don't have a good sequence background or a bank for Blacks. We're starting to see that we do have a lot of von Hippel-Lindau syndromes, which we have never known and tested. You can pick that up in an individual. The good thing with NGS is that I'm doing it for an individual. What Kenji is doing is he's sequencing all your genes. It doesn't matter where the genes are, sequencing all your genes and all your cancer genes and picking mutations and mistakes that are there. Up to now, we don't know what to do with 99% of the results we get. That's the truth. But we do know what to do with some of it. Some of it, we know what is happening, like things like TP53, we do know TP54, we know things that are driving cancers, but we don't have drugs for. But we know certain things. So if I do an NGS and it comes back, let's say you have an EGFR mutation. The question is it a driver mutation or is it a passenger mutation, which is a standby mutation? You do have a BRCA mutation and you have breast cancer. I could see that you are Ashkenazi Jews, but I'm not going to stigmatize. I'm going to be talking about the first part is you have a BRCA mutation. We have drugs that can respond to it. Let's treat you. But there is an underlying thing we must talk about race. I know many people don't want to talk about, we must talk about our differences and in a positive way. Now you're Ashkenazi Jews, you have cousins, or I will try to say, or you have sisters, you have a BRCA mutation. It's good for us to get others tested and so that we can help the others. We usually send to counselors, social workers, that's what we do in the hospital, so that we don't make it more about we get everyone's help. We don't make it more about your ethnicity and stigmatize you, but we try to find solutions because that is not going to go away. It's always going to be there. That risk is going to be there. Being Black, I get people coming, sickle cell, other things. You have to be able to make sure that these patients are counseled in such a way that they have informed consent to make decisions in the future that they would have help from both counselors. I know I'm trying to integrate the last part of the question to what you answered, but we don't generally shy away when we pick it up. Yes. We try as much as possible to talk about it, yeah.
Thank you, Dr. Mono.
Just in the design of clinical trials too, that's where it has to be more inclusive, right? Clinical trials for some of this medication. You can't really be running a trial just here in the US, then you're expecting that the results will be the same when you take that medication, maybe to a place in Africa. So including people from diverse backgrounds, especially in the design of most of these clinical trials or even companion diagnostics, I think it really helps too.
Yeah. And your second question,
I think that was the second question, was really about, because the marker that you're using in the lab, I'm assuming it's coming from the general literature, like the clinical trials, right? To make sure that they're actionable markers. So the question was, how do you actually mitigate the risk that if you give a result to a doctor about a marker that has been validated, how does it actually take that result and interpret it within its own context, knowing that this marker was validated in a clinical trial, which may not have representation of his own population? Is there like some kind of warning or comment, or I don't know, can you just tell us more about how that communication?
Yeah, that's what we are good at. So we have all the disclaimers, right? So at the end of each report, you see tons and tons of disclaimers regarding a test. There are some tests that are straightforward that we don't put a lot of disclaimers, right? And all those things. So when you go, like the results I shared, when you get into the detail, we'll cite the literature that have said that somebody that's having this particular mutation will be resistant to this particular drug. But however, right, there's always that clause. However, it may not be applicable to every patient. So we always say the clinician should interpret this result based off on the clinical presentation of the patient and make the decision. So the way you look at the lab is, we provide you the scientific information. I am not the clinician. I'm not Dr. Foma, and Dr. Foma is not me. What I do is that I do the sequencing. Hey, Oncologists have seen this resistant mutation. And per literature, if you give this medication to this individual, the individual may not respond. That's all. Then you, the clinician, now you take it from there based off on the clinical presentation of that individual to see, all right, maybe I can prescribe this. If it was a one-to-one thing, then maybe we will not even need clinicians. Maybe you just get the lab result and just prescribe direct for that patient, right? But the clinician has to manage the patient. The clinician is not managing the laboratory result. That's one thing. You manage the patient, and at times there's always a difference between the genotype and the phenotype. We've seen this a lot when you do antimicrobial sensitivity testing, where you can have this particular, especially now that PCR is becoming very popular for urinary tract infection diagnosis. So you identify a particular gene, right? That, hey, this person is resistant to vancomycin by PCR. But when you do the antimicrobial sensitivity, where you actually grow the bacteria, put the drug in it, you see that it is still working. Right. So again, too, it's still never a one-size-fits-all, because it also depends on how the clinical trial was designed. And again, too, if you carry this and you are implemented in Cameroon, meanwhile, everything was done in America, it's another challenge. So that's why you don't just take a test and you just implement. You need to validate that test as well to be sure that it works in that particular context. So I'll push it over to the clinicians, to talk more on what they do when they get some of these results.
Yeah, just to add, Dr. Mfuh, before you answer that, I just want you to comment also on the fact that, is it possible that when you get a result from the lab, like the one that Dr. Kenji mentioned, that you exclude a certain treatment based on the result you get, and you put the patient at risk of not getting the proper treatment because he may still be responding to the treatment that got excluded based on the laboratory results? So over to you.
Yes, so I would answer the first part, then I'll go to the research. That's why it's very important. You're lucky to be in Canada and I'm in the US. We're both lucky. Why? Because the US and Canada and Europe, they're very strict. So why are they strict is, I'll take, for example, the Breast Destiny trials that validated trastuzumab deruxtecan, which is a drug we use a lot. Why? Because it was done a lot in Europe, in Asia. Also, the first trials for EGFR were done in the US, but the later trials that changed the world were done in Asia. And the population, they took into consideration the populations that we have there are not the same like our population. Okay, Asians are responding very well to this drug because they have a huge population in China. We can run a clinical trial in less than 5 years. We'll have good results because there are 1 billion or 2 billion people. Now, does that work in Americans? It had to take the Destiny 02 in the US to validate it. They know they have a more diverse country. So US and Canada decided, let's validate it, US, Canada, and Europe, and divide it in sub-regions and see those who are going to respond. And they validate that in almost all trials. When we see a clear pattern that everything that works in Europe is actually in Asia is actually working in Europe, like we're wasting our time, every trial is coming out positive, then they stop and they start validating some of those trials. But they try as much. And in Africa, we are poor. Not that we're poor psychologically, we think about ourselves being poor, but we would have to validate some of these trials because we have the most diverse population in the world. You cannot believe it, but the genetics of someone in West Africa is more different than the guy in East Africa, even when compared to Europe overall, when it comes to polymorphic variation. But people don't know that Africa is the birthplace of humans. Variation in genes, when you take African Americans who moved here in the US, analyze their genomics and compare it with those in West Africa, there is a whole huge gap. They are even closer to white sometimes. So I don't want to go into that controversy, but that tells you how genetic variable we are. So that's something that has to be tested. It's a fact that we have to do that, but are we going to be able? But when we see patterns that show that EGFR, no matter your gene, no matter where you come from, as much as you're humans, we are 99% close to each other. It's working for everybody. Those are some of the things we may not be able to, or we would use more resources to test. Now, your second part of your question is, do we eliminate people from certain medications based on their genes? That's true. And based on the tests we do, that's true. So there are times when I do a test and I figure out, let's say for colon cancer, I'll just take easy example, and you come out your KRAS and NRAS wild type. I know that you're going to respond to a drug called cetuximab and pembrolizumab. If your KRAS mutated, that drug, I can give it to you if I'm just wicked, but I know that there are exceptions, but there are 99% of the case that we have seen in clinical trial that you will not respond to it. So that drug actually is kicked out for your case. I'll go back to the results of the EGFR. The EGFR is an important gene. It's actually HER1. We test HER2 a lot, but EGFR is HER1. Those who don't know, there is HER2, HER3, HER1, HER4. EGFR is HER1. Another way of calling it. We were so excited about EGFR, not me, but those who discovered it in the days. Then they did clinical trials in lung cancer. We thought everyone was going to respond. It was a negative clinical trial. In the early 90s, everyone was baffled. Why? We think this is actually driving cancers to grow. And then someone went to the lab and said, Kenji, can you guys test us? Go back to that trial and test us. Everyone who was involved in that trial, when we started doing NGS, we had the genes were preserved. Can you test for EGFR mutations? Then we saw that, okay, those who had an EGFR mutation, those who were responding. It means the general population was not responding, but it was a subgroup of about 10% who actually had dramatic response. That's why we saw those people really respond. Then they redesigned the trial later on and said, let's test EGFR first, use companion diagnostic. This is just summarizing all what we have done today. Let's use companion diagnostic, get the right people, and then we treat them that way. And since then, precision medicine has become the standard of care in oncology since that same discussion, where we test for EGFR, we test for ROS mutations, we test for NTRK mutations, we test for ARC mutations, we test for more than 25 companion tests that are done, but this now can all be done with an NGS most of the time. At times we do a DNA NGS, at times we add the RNA, at times we add the proteomics, but Kenji is going to tell you this is getting very complex on their end. We are the user end. We are satisfied. We just prescribe things and we wait for them to send them to us. So it's amazing to see that science from bench, that's from the desktop, from the bench is moving right up to the level of the tertiary level and is affecting care. So thanks for the wonderful questions.
Thank you. I just going to have one. Go ahead, Dr.
I'll
Kenji.
just throw in one thing. If you were talking about a lab test and the doctor may not really treat based on that lab test. So we have sensitivity and specificity, right? Sensitivity is the ability to test positive when that pathogen is there. But what the clinician really wants to see is what is called a positive predictive value and the negative predictive value of a test. So you can have a test that is 100% sensitive, meaning that if there is any COVID in this sample, we are able to pick it up. But then when we talk positive predictive value is like, what is the probability that this patient that actually tested positive actually has the disease? That's what's referred to as a positive predictive value. So even though you are positive, what's the probability that you actually have the disease? I'll give you an example. Somebody comes complaining with urinary tract infection. You take urine, then you do a PCR. Understand that dead bacteria will still amplify. That is why even for significant bacteria, we always take 10 to the 5 colony forming unit. So the fact that that person is positive by PCR for E. coli, what is the probability that E. coli is actually causing that disease? So you can have sensitivity of 100, but positive predictive value is probably just about 30%. So in that case, even though it's positive, the clinician will not really trust, right? Like the probability is just 30% chance that you actually have that disease. So those are some scenarios in a laboratory test where, you know, a clinician may not really respect the fact that it is positive and treat based on that. Yeah, thank you.
Thank you, Dr. Kenji. I wonder if you can comment on the application of artificial intelligence in immunochemistry, particularly regarding the whole slide imaging. We've talked about technician skills. Is it an opportunity here to basically extend that skills if it's not available locally, but like in Cameroon, for example?
Huge opportunity. And at Stanford, we already started developing AI for pathology slides. But again, with AI, you need to feed it with a lot of information. So immunohistochemistry is one of the cheapest laboratory techniques that you can implement, right? As compared to NGS, immunohistochemistry is relatively very cheap. The problem is coming to interpreting those slides. And I think this is where AI will really come in. So now, but you need to train the model, right? Like now, I'm not sure how far the researchers have gone here, but at Standford, but now you can now push this slide, right? You use AI algorithm and they're able to identify tumor cells. I think Mayo Clinic is also doing things like that. So with the help of AI, I think it's going to make some of this testing readily available in a lot of places. At Stanford, we now use digital pathology. Pathologists does not have to come to the hospital anymore. They sit in their homes, right? We have scanners. Scan the slide, everything. They just take a look at it, blah, blah, blah. They give their interpretation. Then they can also do consults. Right, consult means that, hey, the pathologist looks at it, but I'm not very sure. Then you now get another pathologist from a different hospital to give a second opinion regarding that slide, right? So AI is going to revolutionize things. It's already revolutionizing the way we look at NGS data all on its own in terms of coming out with all these algorithms. I also give you another example. Microbiology has really gone a little bit ahead. Now in microbiology, we are implementing a robot where, it just dips the dipstick, do the streaking by itself, does the incubation, then it's going to take those plates and read them. Like it can look at just the colony morphology and everything of that particular organism and start giving you some ID, right? But before we got to that stage, you have to feed it with millions of data. So I think one way to overcome some of these challenges is, implementing AI. And again, AI will not take the place of the pathologist, right? Even though you have those AI, you still need to make sure that you put in all those safeguards, right? In terms of quality checks and all those things. But I really see AI playing a vital role here. And I'm trying to look at a world where I would say, for example, in Cameroon and most African countries, testing for infectious disease, I would say it's fine. Malaria and all the like, yeah, our testing is good, but the real challenge comes with pathology, right? Reading those slides and so we can have a scenario where you train the laboratory scientists in Cameroon on how to stain the slides very well. Then we have those scanners where you now stain the slide, you scan your slide, you send it to a pathologist. It could be in Cameroon, it could still be abroad. They look at the slide. You can have people that are volunteering to do that for countries. ASCP does a lot of that in Kenya. They look at the slides, they do the interpretation, and if you have doubts, you send it for consult. That's a good thing. So you have like a peer review community. Like, I'm not sure if it is especially between 1 and 3, right? I'm not sure what's there. Then you send it to another person, they give their opinion, another people give their opinion. Then you have like a group chat just on that particular image. I think it's going to really change things. But again, for this to work, you need good internet connection, and a host of other things to implement this.
Yeah. And just my last words, as you said, there were papers that were presented in ASCO this year and in ESMO this year that even showed that AI, after good training from very reliable pathologists, was able to predict better, especially in testing for ISC in R2 patients, and their models that are being designed. And these papers were presented in ASCO and ESMO. And AI is not here to take our jobs away. It's going to take some, but it's here to make us more efficient, and we should embrace it. And Africa has an opportunity to catch up. 50 years, ISC was developed more than, I don't know if Kenji can correct me, more than 50 years ago technology, 50. But we're still having difficulties even doing one ISC appropriately in Cameroon. So that tells us how we can bridge the gap by getting these models and also implementing them and using telemedicine and other things. So thank you very much. And I think this was a wonderful session. Sandrine, this was really a wonderful session, and over to you for closing remarks. And you can pass over the mic to those here.
Thank you, Dr. Mono. I don't know if Elvis is still with us. If you want to say a few words. Elvis? He may not be on. So I'm not sure if I should close in English or French, but I'll start with French. Merci beaucoup d'être là. C'était un sujet intéressant. J'espère que vous avez appris énormément. Moi, j'ai beaucoup appris. J'ai aussi apprécié l'échange au niveau personnel. Je vais juste résumer en ceci. Dr. Kenji a parlé ici de la médecine diagnostique, particulièrement en soutien au diagnostic que le médecin pourrait poser. On a vu différents types de diagnostics, notamment le séquençage par PCR. On a un peu parlé de pathologie. Mais surtout, je pense qu'il y a à retenir pour ma part, c'est que cette médecine de diagnostic de précision, elle est essentielle en fait pour le patient. Ce n'est pas une médecine de population, mais une médecine individuelle. Et elle permet aussi au niveau financier d'économiser beaucoup d'argent en ayant la bonne cible dès le départ pour le médecin et aussi au système de santé. Alors, Dr. Mono a parlé aussi du fait que c'est vers là que la médecine du futur se tourne. Nous ne sommes pas encore là en Afrique. Par contre, en Afrique, nous avons déjà certains moyens comme les séquenceurs à PCR qui ont été distribués durant le COVID. Dr. Kenji en a parlé. Donc, nous avons déjà l'existant. Et Dr. Mono aussi a mentionné que nous avons aussi de belles têtes, des personnes ingénieuses qui travaillent dans les laboratoires au Cameroun. Donc, il s'agit maintenant d'amplifier la communication autour de ces cibles qui sont importantes pour la médecine de précision. On a discuté également aussi de l'importance ici de sensibiliser les groupes qui font je vais dire advocacy, le mot me manque en français, également de sensibiliser les technologues eux-mêmes, ceux qui pratiquent qui sont derrière les laboratoires au Cameroun. Et enfin, moi, j'ai noté que l'intelligence artificielle va être d'un support énorme, non seulement aux États-Unis et en Amérique, mais probablement en Afrique, où on parle ici d'une limitation au niveau non seulement des pathologistes qui sont bien formés, mais aussi de l'interprétation. Donc, en conclusion, en anglais, thank you everyone for being here. I hope you appreciate this talk. You enjoyed as much as I did. Thank you, Dr. Kenji, for the wonderful talk. Thank you, Dr. Mono, for the interactive remarks. And I wanted to say a few words regarding what has been shared, just to, for everyone. The subject today was a very interesting one because we didn't talk about population medicine, but mostly individualized medicine. Diagnostic medicine is really precision medicine tailored to the patient. That's what I learned, and I hope you also were able to pick that up from the conversation. I also understood that there are a lot of challenges in Africa, but the good news is we are on a good foot, thanks to COVID, since now we have some of the material that is needed to go ahead with precision medicine or diagnostic medicine, meaning the sequencing machine. And we are also blessed in Cameroon in particular with very good lab technicians. Dr. Mono mentioned how good they are at finding ways without means to implement those technology. And I also thank Dr. Kenji for talking about the relationship between the physicians and the lab pathologist or the lab researcher. So they really work hand in hand. That's what I understood. One does his job by providing the evidence, the genetic evidence, and the doctor is actually treating the person as a whole, of course, basing his opinion on what is being delivered by the lab. So I'm going to conclude with this. The future is bright. Artificial intelligence is probably one of the way that future is going to be even more brighter, particularly with expanding the skills of the pathologists in the US and also in Africa. Thank you all. Over to you, Dr. Mono.
Wow, thank you very much. And Sandrine, it's a blessing having you. And today we've been very bilingual and truly Cameroon in the town hall. And we hope to continue this way. I would like to call on Dr. Kenji to give his last words. And if Elvis is back, he can just say a word and then we close the session.
Well, I just say it's been a Very good interactive session. I was having another meeting and I told them I'll only be gone for 1 hour. They were so interested that we've been here for, almost 2 hours, 30 minutes. So Dr. Mono, if they query me, I'm going to send that over to you. So I think as somebody puts in the chat that the future is really bright in terms of molecular diagnosis, especially in Cameroon. I think that's where we should be moving towards. It's going to take some time, but we need to advocate, right? Before COVID, who could have known that we'll be having all these many thermocyclers that we currently have in Cameroon? Before it was like, it's not possible, but now it's possible, right? We've been able to conduct PCR, conduct sequencing for COVID, and now we can better utilize those platforms to do other things. And I always see the town hall as a great place, to network and to partner. If your lab needs any of those, if your lab is considering implementing, PCR or NGS or anything, we have the expertise here. I'm not the only lab person here. There are a lot of other brilliant people here as well that can help, right, in terms of doing those implementations. And again, if anybody thinks creatively, we need to be our own advocate. If anybody can think creatively as well as to how can we put our voices together to make NGS and PCR more accessible, in our community, that would be great. Somebody said the ability for us to produce our primers and probes. Yeah, we can do that. I don't see what can stop us from making our primers and probes. But if you don't tell people, they will not know that this is something that is there, right? So the first thing is education, and that's what the town hall is all about. For us to educate people that, hey, there is something called companion diagnosis, and there is something called NGS that we can potentially implement. Then we now work together to see how we can implement all of this. So thank you all for a great session today. Thanks.
Okay, thank you all. And I think we are waiting. We have a parking lot for about 15 minutes where people can connect and get to know each other.